So I'll start now. Hello and welcome back to Archbold Staff Seminar Series. Today's speaker is Dr. Betsie Rothermel, the Program Director for Restoration Ecology and Herpetology. Before we begin, we'd like to share some helpful tips for navigating the webinar. All participants are muted and your video cameras are off. You may submit your questions using the Q&A button along the bottom of the screen for our panelists to answer after the lecture. Use the chat button to communicate with panelists and attendees. Make sure to choose all panelists and attendees so everyone will see your comment. Thanks everybody, and now I'll turn it over to Dr. Rothermel. Betsie?
Hello, can everyone hear me? Oh, I don't have the chat open.
Yes, we can hear you.
Okay, thanks. I need to do one thing to get the chat open. But not that. I'm so sorry. Okay, I've got my chat. You can see my slides in a second here. Okay, can everyone see my slides? Where did my chat go?
Yes, this is perfect.
Thank you so much. Sorry, clearly I am not an expert in the Zoom webinar yet. Okay, thanks everyone. A little fumbling around there. Thank you for being here. As Laura said, I'm the director of Archbold's Restoration Ecology and Herpetology program. And I've been fortunate to work here as a research biologist since 2008. So in our program, we work on wetland restoration and we study different aspects of the ecology of amphibians and reptiles with a focus mainly on amphibians and long-term studies of gopher tortoises. So I'm going to start by giving a brief overview of the breadth and depth of the nearly 80 years of herpetological research that's been supported by Archbold Biological Station. And then we're going to do like a deep dive into studies of toads, which I think are a fascinating but somewhat underappreciated group of amphibians. It turns out toads have been the subject of some groundbreaking studies dating back to the mid-1940s. So those were the first few years after the station was established by Richard Archbold, who was a research associate with the American Museum of Natural History. So, oh, someone's saying my audio is not working. I don't know how that can be the case, but oh, I'm so sorry. Yeah, it's going to be a problem when we get to the computer sounds. We'll see how this goes. Okay, so herpetology is a subfield of zoology or wildlife biology that's dedicated to the study of reptiles and amphibians. So herpetologists study ectothermic or cold-blooded vertebrates known collectively as herps. And if you watch a lot of nature-focused programs, you might get the impression that most herpetologists study all these dangerous animals. You know, they're out there wrestling alligators, hunting pythons, that kind of thing. But that's really a kind of a biased view, and I think it's mainly driven by the need for high TV ratings. That's not really what drew me to the field. One of the things that drew me to the field was the opportunity to work outdoors and with animals I'm really interested in. So a lot of herpetologists really do spend a lot of time in the field, especially if we're needing to collect also environmental data to look at things like behavior, population dynamics, or interactions between species. However, if I were mainly studying anatomy or physiology or maybe genetics, I'd be spending most of my time in the lab probably. So where do herpetologists work? Well, they can work as faculty members or research staff at colleges and universities, or they can be in government agencies, museums, zoos, or other nonprofits. So we have had a lot of herpetologists come through Archbold over the years. You know, Southern Florida is kind of a herpetologist paradise, given its subtropical climate and the diversity of herps. We also have a lot of endemic species here on the Lake Wales Ridge and other central Florida ridges. So species like the Florida scrub lizard, for example. And as field stations go, Archbold is well known for its facilities. It has a really wide variety of habitats, not only on the core station, but also on our reserve and ranch properties. So you can find everything from xeric scrub and sandhill to prairie and pastures and to seasonal ponds and freshwater marshes. These all have sort of unique communities of amphibians and reptiles to study. I also think people come here because they want to study some unique ecological relationships. For example, people are still having trouble hearing me. Try one thing here. Does it work can people hear me better now? Or it doesn't make a difference? Okay, well, I'll just keep going. So the people are interested in a lot of things like the species-specific adaptations to living in fire-prone ecosystems like the Florida scrub, or they may come to study invasive species ecology. You know, how are native and introduced species interacting, and what are the impacts that invasive species might be having? So we have a pretty good handle on the diversity of species that occur on Archbold. We've documented 55 species of reptiles in 16 families. This includes glass lizards, introduced geckos, anolis lizards, several species of skinks. We have a large number of species of colubrid snakes, like this yellow rat snake. We have 3 species of venomous pit vipers, and we have several species of both terrestrial and aquatic turtles. And then with the amphibian diversity, the fauna is not so diverse. So we have about 23 species in 9 families. We have a lot of pond breeding anurans, including toads, tree frogs, and the true frogs, like this gopher frog in the ranid family. And we have several interesting fully aquatic salamanders, sirens, like shown here, and amphiumas. But in general, we don't have many salamanders here this far south in southern Peninsula Florida. If you really want to find a lot of different kinds of salamanders, you need to go up to cooler mountainous areas like the Southern Appalachians. So here's a slide I had fun putting together showing the diversity of herpetologists that have worked at Archbold. I sort of arranged chronologically from left to right. This is not everybody who's ever worked here. This is, we've got Charles Bogert, who is curator for the Department of Herpetology. And I'm going to come back to him. He was curator for the Department of Herpetology at the American Museum of Natural History. So I'm going to come back to him shortly. We have Dr. James Lane here. He was the station's first research director. He was a broadly trained zoologist who studied herps as well as small mammals and birds. And he worked here from the late 60s all the way through the 80s. I love this photo of him holding a gopher tortoise standing next to Richard Archbold, our founder. And then from the 90s till through today, we've had, just hosted a lot of researchers from different universities. Kim Babbitt did her graduate work at UF, University of Florida, and worked on Archbold's amphibian communities at Buck Island Ranch. Earl McCoy and Henry Mashinsky from the University of South Florida have done a lot of studies on gopher tortoises and sand skinks with their collaborators. And then, on through today, you'll see there's a kind of a common theme running through here of gopher tortoises, so that they have been a big focus of a lot of research. You might also notice that if you look at the slide, step back, that there is a pronounced gender bias in the slide. So like most STEM fields, herpetology has traditionally been male dominated. However, if you were to attend a herp scientific conference these days, I can assure you that it would not be nearly so skewed. You know, if there's any young women or other members of other underrepresented groups who are watching this, if you have an interest in herpetology, you are welcome in this field. And I want you to go here to check out the Herpers Twitter feed for stories about you know, what women herpetologists are doing all around the world. So thanks to all these people and many more who I couldn't fit on that slide, we've had a little over 200 herp-focused research papers published, including about 162 studies in the southern Lake Wales Ridge region. So this includes studies conducted by both in-house and visiting scientists in and around Archbold, including Buck Island Ranch, which is located just off the ridge to the east of the station. As you can see, most of the studies have been conducted focused on reptiles, and there's been a big spike in that actually the last decade. This category called both essentially is studies of multiple species. So studies that weren't just focused on a single species. And along with publishing, many researchers also collected specimens as they were doing their research. We maintain a collection of about 2,600 alcohol preserved herp specimens. About 86% of those are from just these 6 counties here in South Central Florida. People would collect specimens for a variety of reasons. So if you find a new species, well, maybe not a new species, but a species in a new area where it's never been documented before, you might want to preserve it as a voucher to you know, make sure that later on people might have questions about its ID or something like that. They could look at the actual animal. Sometimes you have to sacrifice your animals to get the data you need. So you have to, like, if you're doing a study of dietary you know, characteristics, you have to look at the stomach contents, but then you might deposit the specimens in a collection so that future scientists could learn other things from those specimens. Unfortunately, a lot of these specimens also, especially of the snakes, were roadkill. But if they were pretty intact, then the researchers went ahead and deposited them in our collection. Okay, so as I said, we're going to talk about toads. There are 3 species in the family Bufonidae here in peninsular Florida, but worldwide there are over 600 species in this family. People are often wondering what is the difference between toads and frogs. In general, they're in the same order, the Anura, so they share a lot of common characteristics. But one thing is that frogs tend to be stouter bodied and have relatively short legs. So think of these as more like hoppers, not leapers. Also, the obvious thing is they have this really bumpy skin with what we often call warts, but these are not warts. What these are little concentrations of specialized granular glands that produce substances that are distasteful or toxic. So they're producing chemicals that defend them from predators. And toads have this huge parotid gland behind the head that produces bufotoxin. So they bufotoxin, specific chemical in their skin. All of the native frogs and toads in Florida have a typical life cycle where adults lay eggs in water, then the eggs hatch into these aquatic tadpoles, and then eventually they metamorphose into terrestrial juveniles and grow to be adults. The thing about toads is that they have incredibly rapid larval development. So some of these species only take 3 or 4 weeks to go from hatching out of the egg to metamorphosing into a terrestrial toad. So that means they can take advantage of some bodies of water that don't hold water for very long. And both of the photos on this slide are of the southern toad. And that's the first species I was wanting to tell you about. So this is a common native species in the southeastern US. It's a medium-sized toad, maybe 3 or 4 inches max. It breeds in ponds and ditches from March through October, and a female toad can lay about 4,000 eggs in these really long strings. They are active mostly nocturnally, so you can often find southern toads in residential areas. So if there's a street light that's attracting a lot of bugs, the toads will be down there picking off the bugs, ants, beetles. They also eat snails and other invertebrates. So I know if you notice from these photos, they're really variable in their color. So that's not a good way to tell this species apart from other toads. But a good trait to use is to look at the top of their head, and they have a pair of cranial crests, so like little knobs on the top of their head. So that's a good trait to identify southern toads. It turns out that the southern toad was the subject of the very first amphibian study conducted at Archbold 76 years ago. It was a field study of homing in the Carolina toad by Charles Bogert. And they were calling it the Carolina toad, but its name has changed. Common name and scientific name have changed since then. If Charles Bogert's name rings a bell, it might be because you have this classic CD in your collection, which I happen to have because I'm a big nerd. So this is the biological significance of voice in frogs. So he was really interested in vocalizations of amphibians. He made recordings all over North America of different species in the 50s. And then he produced and narrated this wonderful collection. And so what I want to do is play you his recording of southern toads. So take a listen, and I hope you can hear this.
In some species, the air is rapidly forced back and forth over the vocal cords at approximately 75 times per second to produce a high-pitched trill of the southern toad, Bufo terrestris, for example.
Okay, let me play that again. Is that coming through, that nice long trill? That is the musical call of the male southern toad. And they're calling right now. I live on a little lake here, about 15 minutes from Archbold, and they've been calling a lot. We've finally been getting some rain. So this homing experiment they did was interesting. If you're familiar with Archbold, you'll recognize this is our main research building here, and you're looking west out into the scrub. There was a big wetland here, actually still is, and it was a breeding site for southern toads. And Bogert was here in the summer of 1944, and he decided to investigate what is the maximum distance a toad is capable of traversing to return home. So if you displace them out some distance away, how well can they orient and get back to their little home territory? And there was a high concentration of toads right in around the buildings and around a garden area here. So he would just go out every night from 8 to 1 in the morning, do a couple of loops around this area and catch as many toads as he could find. And over the course of that summer, he released toads in groups at different distances and in different habitats away from the main buildings. And then he would patrol that area again at night and look for any toads that returned. So he had marked them, and he could look and see if it was one of his toads. So what did he find? Oh, and I meant to mention, that farthest site, you might have missed that, it's way out. It's actually west of these buildings, out a mile. Okay, so that was his most distant site. Okay, so he was able to recapture about 26% of the 200 toads released. They came back, he captured them, and as you might expect, the ones that were released closer to the buildings, there were more of those recaptured. At least though, eight of 43 toads that he released a mile away returned. And there was one toad he released 850 yards away that came back within 1 day because it was a rainy night and it just was able to do one big movement back. But you know, so that was interesting. It showed they could really orient and navigate back to their place. How did they do that? He thought it was unlikely that the toads released to the west. If you noticed in that photo, it was really open and it had been logged recently. And so he considered that to be pretty unsuitable habitat. He considered that any toads released out there could not have been familiar with that area. So they weren't navigating back by like known landmarks or anything. And he suspected that they were using the sounds of the chorusing male toads there at that breeding site to orient and find their way back. What's neat about this is that this issue of the homing mechanisms in amphibians is still really a puzzle for herpetologists. And this study by Bogert, published in 1946, is still cited on a regular basis. So I found papers even as recently as last year citing that study. Here's one from the Journal of Experimental Biology. And these authors did a similar displacement experiment, but with a small poison frog in the Amazonian rainforest. And they found that even when you displace these frogs over 500 meters away, they showed very directional movements back. And 10 of the 12 that he released returned within 13 days to their own territory. Now think about an Amazonian rainforest. Could they be using visual cues to do that in a dense jungle? How would olfactory cue? Could they smell their way back? This paper was not conclusive. It's really a hard question to answer. One thing that we have learned though from other studies since Bogert's time in species like bufo toads in Europe is that other cues that amphibians can use include things like celestial cues if it's a clear night, or they can, some of them, use magnetic cues. So they can sense extremely slight variations in the Earth's magnetic field. And that kind of tells them where they are relative to where they want to be. So basically from studies like all this collection of studies, we know that amphibians use multiple sensory mechanisms to do this. But for most amphibians, this is still really not well studied. So the oak toad is another of our common native species. It is maybe a little more specialized in its habitat preferences versus the southern toads. It's most common in scrub and pine oak forest or sandhill. These are really small toads. They're only about an inch and a half long. They breed in ponds and ditches from April to October, and they lay up to 500 eggs in kind of short little strings. One thing I think is neat about them is that they are most active during the daytime, which is impressive when you think about how hot it gets out in scrub and the fact that amphibians have a moist, permeable skin and they're constantly losing water through evaporation. So how is this little toad able to hop around without just dehydrating out there in the scrub? One of the tricks that toads have is they can store a lot of water in their bladder as dilute urine. And then as they start to get dehydrated, they can reabsorb some of that water. So they're basically, it's like they're carrying around a little water tank. And then if they run out of that, they can use their hind legs and dig down and cover themselves in the soil, in the substrate, so that they stop losing water through evaporative water loss. So that's one of their interesting talents, I think. So the oak toad, like the southern toad, mainly eats bugs, ants, beetles, other small invertebrates. And I wanted to play you Bogert's recording again, this time of the oak toad. Turn up your speaker.
A much smaller toad, the oak toad, Bufo quercicus, has a totally different voice consisting of a short, high-pitched whistle.
Okay, were you expecting to hear that? Anybody, does that remind you of any other animals? A lot of people think that they sound like baby chickens. So I want to tell you about some of the research on oak toads. They really didn't get much attention historically, but I've had a couple of interns pursue independent research on them in the last few years. And one was Clay Noss, who was here as an intern in 2012. He looked at the growth and survival of the tadpole life stage in ponds that had different times since fire. So you might not think of ponds burning, kind of a strange concept, but the ponds in the scrub, most of them are just shallow depressional wetlands that are dry for most of the year. And so when a fire comes through in the dry season, it burns through the vegetation in the ponds as well. And that actually can change, have kind of a short-lived effect, but it can change the water chemistry when the ponds refill. So the ash from the fire raises the pH, makes it less acidic. It releases phosphorus. The fire, burning of the vegetation releases phosphorus, so there's some nutrients made available to the pond. So we thought this might make a difference. And sure enough, if you look at this graph, we had more survivors in these enclosures. We just stocked these enclosures with tadpoles. We had more surviving tadpoles in ponds that had been burned within 4 months, compared to either ponds burned 3 to 4 years ago or ponds burned, long unburned ponds, like more than 11 years. And the tadpole survival was positively correlated with pH. So it seemed like they were benefiting from the higher pH, lower acidity in the ponds. So just, it's not surprising maybe because these oak toad tadpoles are as a species, the species is adapted to these fire-prone areas. And here it would be interesting to know, for example, if the adult oak toads, the females preferentially lay their eggs in recently burned ponds, like there would be a good follow-up study right there. And then 2 years ago, Madison Harmon was a research intern in our lab, and for her independent research, she was interested in exploring the ability of oak toads to match the color of the substrate that they're on. So, a lot of reptiles and amphibians can do that. And so she did some really nice experiments where she acclimated the toads to like a neutral gray background. And then she put them either on black sand or white sand. And she tested each toad on both colors so she could look at its full range of ability to change color. And you don't even have to really look at graphs to see that they are really good at this. So here's one, like this one that turned almost completely black. If it weren't for that light colored mid-dorsal stripe, you really wouldn't even see that little toad. The other thing Madison noticed was that about 6% of the oak toads around here have this really washed out color. They don't have any of the black spots. So there's sort of this unspotted color variant in the species. And based on a search of over 400 records in iNaturalist, if you're familiar with that app, it looks like this is restricted to Highlands County, Polk County, and Charlotte County. So we don't know much more about that, but it would be interesting again to do follow-up studies. So for example, maybe we need to establish whether there's a genetic basis for this. It's not just a phenotypic difference, like there's, some are genetically this way, and therefore it's a heritable trait. And what are the selective pressures that allow this unspotted morph to persist? So for example, are certain predator, is one color better at blending in certain situations? And that's how you can maintain this polymorphism in color within a species. Those are all questions that arise from observations like that. So now I'm going to switch to the cane toad. If you live in Florida, though, you might know these as the bufo toad. A lot of people call them. They're not from here. Let's just clear that up. They're native to Central and South America. They were introduced to Florida in the 1930s through the 50s. There were multiple introductions. They have also been introduced to Australia, Hawaii, and Guam. Anywhere that sugar is grown, because the point of the introductions was that they were supposed to eat these beetles that were damaging to sugarcane. I don't think that worked out too well, but they established and did very well where they were introduced. These are huge toads, 4 to 6 inches. So any toad that you find in Florida that's greater than 4 inches pretty much is a cane toad, not a southern toad. So you can, just the size alone should tell you the species difference there. They breed in a variety of water bodies, they're not too particular. In March through September, a female cane toad can lay up to 30,000 eggs, but that's dependent on their body size. And they eat just about anything that will fit in their mouth, including native frogs, lizards. If you leave cat food or dog food out overnight, they'll eat that. But the big issue with cane toads is their toxicity. So look at the size of this parotid gland on this toad here. They produce copious amounts of bufotoxin that is, can kill a dog, for example. And there's definitely every year instances, unfortunately, of dogs getting hold of one of these toads and ingesting enough bufotoxin for it to be fatal. I guess it has a digitalis-like effect on the heart. So it's serious protection there for these toads. And by the way, every life stage, including the tadpole of this species, is toxic. So we get articles regularly, like this article from a year ago, neighborhoods in Florida that are overrun with cane toads. So during the time of year when they're metamorphosing and all the little toadlets are out, dispersing, people kind of freak out. And they even clog the pool filters. So there's companies that, around who will come and remove cane toads. I think I forgot to play their call. So I'm going to go back. I meant to do that. All right, we need to listen to the call that they make.
However, toads with their less varied calls serve better to illustrate the rough correlation that exists between size and the pitch of the call. The giant toad, Bufo marinus, has a low-pitched trill at a frequency of only 638 cycles per second.
So what Bogart was getting at there, in part, was like that there's a correlation between the size of the toad, the body size, and the pitch or frequency of the call. So the bigger the toad, the lower the pitch. And if you didn't recognize that amphibians calling in the background of that, don't worry. It's probably because you haven't been to Panama. Those were Tungara frogs. That's where that recording was made. Okay, so I talked about some of the human and pet impacts of cane toads. Of course, we're also worried about their ecological impacts, which haven't been studied enough in Florida. There was a study by Mashaka and Powell in 2010. They looked at the diet of cane toads and southern toads where they co-occurred in a residential area near here. And they looked for a whole year, what all were the different invertebrates that each species was eating. And basically they were all eating pretty much the same thing. Some differences in the proportion of prey that they were each taking, but there was strong dietary overlap. So that means that there is potential for negative competitive effects if the food were to be limited certain times of the year. And there is, I guess, have been some declines of southern toads in urban environments, presumably driven mostly by habitat loss, the loss of wetlands. But this would be like an added stressor to have this huge other toad competing with you for food. But the full impacts to native species, its predators, prey, competitors, are not well studied. You know, in Australia, they don't have any native toad species. So when cane toads showed up, the native predators had no, you know, innate avoidance behavior. They would eat these cane toads and just die. And there were documented declines of lizards and snakes and other groups because of the arrival of cane toads. And then you have these other interesting scenarios sometimes. So this, I walked out of my yard one day. I thought, what's going on here? So let's look at this toad here. That small one, that is a male southern toad. He thinks that he has found the biggest female toad to mate with. So he's amplexing her. But unfortunately, that's a female cane toad. So that's not really going to go anywhere. Yeah, and cane toads are expanding their range in Florida still, which brings me to the last study I want to tell you about. This is a study done by a PhD student at Cornell University named Cinnamon Mitten. She's a student in Kelly Zamudio's lab. And she was looking at the expansion of cane toads and wondering how it is that they're able to persist north of Tampa. Central Florida is the coldest place that cane toads are found anywhere in their range. And it was thought that initially when they were introduced that they'd be really restricted to southern Florida. So she collected toads from the original, introduction area down near Miami. And then she collected some cane toads from the northern edge of the range near Tampa. She brought them back to the lab at Archbold, and she did experiments to measure their cold tolerance. And specifically what she quantified was something called CT-min, which is critical thermal minima. So that's the temperature at which an organism loses motor response, and thus the temperature at which the individual would die if, for example, a predator came upon it. So sort of its inability to move and right itself. And this is a quote from her paper, and you see who she's quoting here: Cowles and Bogert, 1944. Hey, there's Bogert again. So that's cool. He also was really interested in physiology and did a lot of work in physiology of amphibians and reptiles. So what Cinnamon found is that the toad populations from Miami were less tolerant of cold than those toads from Tampa. They could not handle the lower temps. So that implies there has been selection for cold tolerance over the generations that cane toads have been in Florida and have been expanding their range. So there's evolution happening there and adaptation. And in Australia, there's a lot of researchers looking at that too, and they're finding interesting ways that cane toads at the edge of the expansion range are changing in physiology or locomotor ability and things like that. So kind of an impressive toad right there. So I hope that little summary of just some of the research that's been done has enhanced your appreciation for the amazing abilities of toads. And not only that, but the kind of insights that you can get from experimental research conducted at field stations. So I'm going to wrap things up with a couple more thoughts about the breadth of herp research at Archbold. And then we're going to end with a little quiz. I don't think I mentioned there's a quiz, but there is, so you need to stick around for that. So after 76 years, you're probably thinking, hey, surely all the herps at Archbold have been studied, right? Not quite. So some species have gotten a lot more attention than others. In particular, listed species, the ones that are considered protected and designated as listed by state or federal governments, only comprise about 13% of our herp community, but they've been the subject of nearly half of the published studies that have been done. So we have had about 63 papers on just 4 species: American alligators, gopher tortoises, eastern indigo snakes, and sand skinks. And likewise, introduced species also get disproportionately more attention. So they only comprise about 10% of the fauna, but they have been the subject of almost 20% of the published papers. And again, I think this is just the interest in them as sort of model species to study these adaptation evolutionary questions, and then also just trying to understand, you know, what is the impact that they're having in their new ecosystem. But this leaves a lot of species kind of unstudied, at least here at Archbold. Here's just some examples of what I'm calling neglected species. And some of these have been very well studied in other places, but the thing is that there could be differences in their life history here in the southern part of the range. So even if they've been well studied elsewhere, we could still learn a lot by studying populations down here. And you know, it's kind of understandable that some of these haven't been well studied. Some are super good at hiding from us, like smooth earth snakes, little fossorial species. The first record of a pinewood snake, Rodinia, wasn't made at Archbold until 2014. We didn't know these were around. There was a visiting Cornell course that found one on the way south end of the ranch. So there's a new addition to the species list for the station. All right. So clearly, I don't have enough careers to study even half of these species. So I hope that people will keep coming to Archbold and tackling some of the questions around these species. Now, here we go, the quiz. I know you guys are ready for this. So you got to hear some of the toad calls. So I'm not giving you an impossible task. We're going to listen to a couple of more of Bogart's recordings. First, I'll start you out easy with a recording of two species made in 1957, west of here along the coast in Charlotte County. Okay, so let's listen and see. And if you can guess or you have some ideas about what you're hearing, type in the species in the chat. Let's see how people do here. Okay, I think we have some knowledgeable people with good ears. Does anybody want to hear that again? Let me play it again, and I'm gonna see if I can find it. For those of you who haven't tuned your ears yet. Oh, that's not what I meant to do. Go back. Okay. That high trill. That's the southern toad. In the background, that whack, 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 those are squirrel tree frogs. I don't think anybody would guess those. They have kind of a nondescript call. Cuban is another, you know, I'd have to look to see if someone suggested Cuban tree frogs as a background. They have a very similar call to the squirrel tree frogs. I would not be confident telling them apart, to be honest. Okay, so are you ready for the real challenge here? This is a breeding chorus recorded in 1954, half a mile south of Archbold, and there's at least 5 species calling. Here we go.
As many as 200 or 300 voices may be heard in a single pond in parts of southern Florida, where the barking frog breeds along with other tree frogs, toads, and frogs.
Okay, yeah, I'm looking at the chat here. There's like a party going on my other monitor with guesses. Okay. Yeah, oak toad for sure. Gopher frog. Yep. The snore. Did anybody guess? He kind of gave away the barking tree frog. So I gave you that one. Let's listen to it one more time. And I'll try to point it out for the people who are having a hard time. Because it really is a thing where you have to tune your ears.
As many as 200 or 300 voices may be heard in a single pond in parts of southern Florida, where the barking frog breeds along with other tree frogs, toads, and frogs.
There the gopher frog, high pitch, oak toad, the pine tree frog. In the background, barking tree frogs, and there were cricket frogs in there too. There usually are. A little just clicking sound that really, yeah, if you didn't know, you'd think it was an insect. So in the CD, Bogart says there were a whole bunch of other species calling in that same chorus, but they just didn't get picked up by the mic, so I couldn't hear them. Somebody asked which frog sounds like the Morse code. I think that's the Pinewoods tree frog. It's like tick, 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 tick. Yeah, they were calling in that clip. So yeah, y'all did pretty well. If you've never experienced in person one of these breeding choruses when the wet season kicks in June, July, it's unbelievable. And I can tell you that oak toads will break your eardrums. So, okay, I guess that is it for my presentation. So I will say thank you for joining in, and we can do some Q&A. Try to answer some questions.
Okay, Betsy, we've got a question about Madison's experiment. What kinds of experiments would you do to study physiological variations in toads like she did?
Well, I thought that was a really clever way to approach it. And it actually took a lot of thought to do that. So, you have to control for a lot of things and come up with a way to quantify how you're rating the color difference. And she did that with photos and looked at the red, green, blue scales. So yeah, I think we followed a pretty typical protocol, I would say, for that experiment of just putting them on different substrates. It's not like they're going to change to purple if you put them on a purple background or something. There is a limit. So in this case, it was more just their background color changing from light to dark.
Okay, we've got a question about, so cane toads have been observed to eat dog food or cat food that sit out, and that doesn't move. So is that something that might be different between them and a native species?
Oh, that's a good question. Maybe. I've never heard of southern toads eating pet food. Maybe they're more, maybe the native toads are more visual predators and cueing in on movement to grab, but maybe cane toads also rely more on olfaction and the smell. And I think they've been known to eat carrion, so scavenging, because you find some really weird stuff in their stomachs if you do diet studies.
Do you know if in those diet studies, have they seen that the toads can eat like snail shells?
Yeah, I think snails were mentioned. That would be a good source of calcium too, in digesting the shells of snails.
Okay, and then do we have any spadefoot toads on Archbold property? How far south does their range go?
Oh, spadefoot toads. I really want to talk about them, but I didn't want to make this an hour-and-a-half presentation. So spadefoot toads, the eastern spadefoot toad, has not been ever documented at Archbold. The closest records for them, though, are close. So you can go off the ridge to the west toward Arcadia and DeSoto County, I think that is, and you pick them up there in large numbers. So they're not like super rare. They're also about an hour north of here and near the Avon Park Air Force Range. There's been 2, I think, seen on the bombing range ridge, which is weird also, considering there's been a lot of studies there and they've only ever seen 2 spadefoot toads on the whole bombing range. So I don't really know what their deal is, but I'm not sure how far south they go, if they go much further south than DeSoto County. I'd have to look at the Florida Museum of Natural History has a nice, you can click and look at their Florida Herp Atlas. I don't know how up to date it is, but it has some range maps for species like that.
So Harrison asks, what's your current research on on frogs and toads, since his research is now completed?
Hi, Harrison. Well, I actually did a smart thing, which is that I cut off sampling for the biopond study. We've been sampling 30 ponds across the station for 3 years or so, and I decided we had enough data and I was going to kind of not keep collecting data. So we're trying to find the time now to dig into that and analyze it and see what we found out. But so I don't actually have any projects on amphibians going on right now. And I'm feeling a little bit of withdrawal, I have to say.
Do we have narrowmouth toads on the station? Yes, we do.
Sure do. I don't think they're super common in the scrub wetlands. Where I see them more in abundance is actually on the reserve or the ranch, where there's a lot of ditches, so almost an agriculturally modified landscape. They seem to do really well out there, and there'd be huge choruses of narrowmouth toads in ditches during the wet season.
Sorry, I was typing. And then we have just some really fun random herp questions if you have the time to answer them.
Okay.
Okay.
I will give it a shot.
Do you have any recommendations for getting rid of cane toads on a property?
So the problem is that if there's any water nearby and there's like, they like open areas like lawns and golf courses and kind of these human modified landscapes, like they like those kind of landscapes. And so when there's this much water, if you're in Florida, it's pretty hard to get rid of them. They're still not very much colonizing natural areas, which I guess is the good story there. But they really seem to prefer these somewhat human modified landscapes. You can collect them basically and humanely euthanize them. And there is a good website at UF IFAS with instructions, but basically to be kind to the toads, but get rid of them. You can use Oragel. If you're familiar with the toothache gel, it's 20% benzocaine. That is an anesthetic. When you rub it on their belly and work with any amphibian actually, they will absorb that benzocaine through their skin and it will put them to sleep. It takes a little while. I've done this with big cane toads. It's not like 1 minute, it's 10 minutes or so, but they basically go down. And then at that point you can put them in the freezer and they will then freeze and die. But you've done it in a humane way where they were heavily sedated so they didn't feel the pain.
Okay, we have a really good question here of how would you explain why frogs start calling when it rains?
Oh yeah. No, this is something that Bogert and other people have written about a lot. So in addition to these advertisement calls or mating calls, certain species like pinewoods, tree frogs, usually it's often tree frogs, just under the right conditions, if it's super humid out or there's I think one of the explanations is that barometric pressure changes associated and humidity changes associated with fronts coming through, bringing rain, stimulates them to just call. So these are calls called rain calls. The frogs may still be, up in a tree. They're not at the breeding pond, and they just start talking. I have no idea why they do this. I don't think anyone really knows. Is it just that it's just stimulating the right it's just like a physiological response when the thresholds are met for humidity. But I suspect there's more to it than that, but it hasn't really been figured out.
Awesome. Let's see. Do you have any comments you'd like to share with everybody about the conservation status of snakes on the station, such as the short-tailed snake or maybe the pine snake?
The short-tailed snake, and what was the second one?
Pine snake.
Oh, Florida pine. I wish I could tell you something about their status. So I have yet in 11 years to personally see a short-tailed snake. I've never seen one. I'm not saying they're not here. I haven't actually looked very hard. I really don't have any active monitoring or research with snake species. And it really takes dedicated effort and the right techniques if you're going to try to monitor snake populations. They're one of the most difficult herp species as a group to get good estimates of population sizes for, because your recapture rates for snakes are generally so low. You can't do traditional mark recapture techniques to estimate population sizes. So we don't have any of those studies going on. We do see pine snakes, 2 or 3 a year or more. So yeah, we see a handful of pine snakes every year. They're super cryptic. So I don't know. They're still around, but I have to say it seems like we almost see more eastern indigo snakes than we see Florida pine snakes. So that's all I can really say about those species.
And then Elizabeth wants to know, what's the favorite herp that you've found at the station?
I have to have one favorite. Oh gosh. I have to say gopher tortoises have definitely become a favorite the more I have gotten to interact with them. So I don't know, amphibian wise, I think I'm partial to gopher frogs too. Yeah, I like gopher frogs a lot. And it's fun because I get to see them not just in the ponds, but then when I'm tracking tortoises or things, we see them in the tortoise burrows too. So that relationship is just really cool.
Yes, cool. Okay, and then we only have one more question, and it's an unrelated question, but whatever you want to provide. Somebody was wondering if you have any information about the history of the introduction of the curly-tailed lizard.
Off the top of my head, not really. I sort of remember maybe they were introduced in the Palm Beach area originally. It was maybe the first point of introduction. They made it to Lake Placid a while ago. So they're here in the interior. But I don't know the whole story. There's some good papers and books that Walter Mashaka has authored, because he really looked at the history of these introductions of invasive species. So I would look for those. We've had one record of a curly-tailed lizard on Archbold. The record consists of a video, because we had game cameras out at gopher tortoise burrows, and a curly-tailed lizard shows up. But we haven't caught them. They're pretty quick and hard to catch. So yeah, that's all I can say about those. But anyway, well, thank you all. Thank you all very much for tuning in and all your good questions. And I saw that a bunch of my former interns tuned in, so that's great. Thank you guys. Good to hear from you, and hope everyone stays well.
Okay, thanks so much, Betsie. And thanks everybody again for attending today. You can join us next week on Tuesday for Archbold's Discovery Classroom when Dustin Angel takes us to a rosemary bulb. And next Thursday, Archbold interns present 2 seminars for the price of 1. First, agroecology intern Juan Olivares presents gas, grass, and biomass, correlations between treatment, plant traits, and GHG emissions potential in abundant species of grasses at Buck Island Ranch. And then Megan Selva, the Jill Abrahamson Memorial Environmental Education intern, presents Persevering Through Change, Transitioning from In-Person Field Trips to Virtual Field Trips. So we hope to see all of you there. Thank you so much again for attending today.